Okay, so welcome everybody to this seminar today. I had the pleasure to introduce our speaker, Dr. Panagiotis Kastritis. Panagiotis or Panos obtained his undergraduate degree in biology from the National and Kapodistrian University of Athens in Greece. He then earned his PhD in chemistry from the University of Utrecht, the Netherlands, working with Alexander Bonvin on modeling protein interactions. He was trained as a postdoctoral fellow at the Pyruvate Center for Molecular Research at Utrecht, again, with Alexander Bonvin, and at the EMBL at Heidelberg, Germany, doing proteomics work. Since 2018, Panos is a junior professor at the Center of Innovation Competence Halle in Halle, Germany. And Panos is a specialist in computational structural biology and cryo-electron microscopy. His research focuses on unraveling the structural function aspects of metabolic pathways in particular, but also other important cellular processes. His work combined structural data from Cryo-EM with data on cross-linking mass spectrometry and other proteomics techniques to model large, multi-component, heterogeneous, and plastic protein assemblies that he tells us. He investigates these molecular organization in native cellular extracts, which is apparently an important emerging trend in the field. So Panos will tell us today about Cryo-EM and artificial intelligence, a marriage made in cellular fractions. So Panos, the Zoom floor is yours. Please, you can share.
Yeah, Susanna, thanks really a lot for the very kind introduction. Really nice. It's perfect that I didn't put also slides to introduce myself. So Susanna said everything about me. So thank you. And thanks a lot for the invitation.
Welcome.
So basically, I'm starting with the motivation of why we are doing this, right? So when we look at a cell with a tomogram here that you see from Yulia Mahamid from EMBL, she acquired it, you can see all of these electron-dense particles inside. And then we can appreciate some structures there inside the cell. For example, the nuclear pores at the nuclear membrane, the ER, ribosomes, actin and tubulin filaments in the cytoplasm, as well as inside the nucleus, we can see chromatin. So we have reached during these last 5 years in a state where we can directly look at a cell with cryo-electron microscopy and see the most abundant species there directly in the cell. But as you can see here, there are 5 of these biomacromolecules modeled, 5, 6 of them. And we all know that the cell is composed of many, many more. And that's why, as structural biologists, we want to understand these in atomic detail. And we are on the other edge of what this tomogram shows. So what we do is that we take each protein, either isolated from the endogenous source in like very high purity, or overexpress it and solve its structure with X-ray crystallography, NMR spectroscopy, or single particle cryo-EM. And of course, this is like how we understand from the structure, we understand function. However, we should not forget that by understanding this kind of structure function, we understand it outside the cellular environment. Below, I show you a protein-protein interaction network where each dot represents a protein molecule and each of these connections an interaction. And therefore, you can appreciate that the task that as structural biologists have to determine the structure of a protein or a protein complex one by one is something that is not going to be solved now, but we are working on it to understand more and more in the future. So basically, we have in situ or in cell structural biology that is emerging that can see structures of individual molecules of extremely high abundance, the ones that we know how they look like. And then we have our in vitro structural methods to understand how a specific protein of interest looks like. What I am doing in the lab is something in between. I am trying to bridge this in situ structural biology and the traditional structural methods that we have by studying cell extracts. So we have an organism of interest, we homogenize it, and then we take the cell extracts and we try to understand there how is the architecture. And this is important because not only proteins and protein complexes might have additional subunits that you might lose in downwards purifications, but also these samples, because they are cell-free, they are accessible to all physical chemical assays like proteomics studies, cryo-electron microscopy, because now you have a thin sample, and of course computational modeling from the data that you are acquiring. And also what is very important is that cell extracts are expected to retain principles of cellular organization. We can all think that cell extracts were used to decipher the genetic code, right? Or cell extracts are generally used for biotechnological purposes to produce certain molecules of interest. So they retain a certain function there. So these are the first images that I acquired, as Susanna said, during my postdoc in EMBL, where I was doing this kind of visual proteomics by combining cryo-electron microscopy with proteomics under the supervision of Martin Beck and Anne-Claude Gavin. And what you can see here is like Cryo-EM images of cell extracts, where you can see this kind of higher order structures. So this is like a hamburger-shaped molecule here. It's called fatty acid synthase, and it's very frequently close to other mega-Dalton biomolecules forming this kind of communities. And to make things short, what I did here is that from such an extract, I could reconstruct the Coulomb potential map of the fatty acid synthase as a community member of this kind of higher order assemblies back then 5 years ago at 4.7 angstrom. And this kind of work with a lot of other data that we're including points to this kind of organization of protein communities that are basically like proteins that are part of the same metabolic pathway, for example, that they are in proximity. Some of them might form multimers inside the cell and organize on scaffolds or on membranes or on structural elements, forming this kind of higher order structures that we see in the cell extracts. And this is in contrast to the fact that we know from our textbooks that proteins are in like this kind of pathways, and the substrate and the intermediate that is being as a product from one enzyme goes as a substrate to the next one, and this kind of linear kind of fashion of proteins working, it's contrasts how these kind of communities are. So here is another image that is the first image that I acquired here in Halle, where I'm a junior professor for cryo-electron microscopy. This is a negatively stained image, and you can see here this kind of cell extract complexity. You can see very nicely with high contrast, because we use the stain here to understand what is there, of different molecules, like here again the hamburger shape, fatty acid synthase, but also a lot of other macromolecules forming this kind of higher order assemblies together. And this kind of samples beg for questions, like, for example, how do we discover and define these protein communities and their members? How do we correlate protein community identity with the protein structure? Because with electron microscopy, we see structures, but we don't know what they are. So we need to use other methods to tell us what we have in our samples. And also, how do we understand activity? So how do we know that our cell extracts is not artifacts, but they have inherent function? So these are the questions that my lab is interested in. And in particular, this is the lab workflow that I have established from 2018 when I came to the University of Halle-Wittenberg here in Germany. And what we do is that we start with our model organisms, Hitomium thermophilum, a thermophilic fungus, yeast, and from here we are isolating different kinds of samples like soluble and membrane extracts. And then we are doing our methods like with our in-house glacious microscope. With collaborators, we are doing mass spectrometry to get proteomic data and also computational structural biology methods we have in the lab. Since, as Shoshana said, I did my PhD in developing the software Haddock in Alexandre Bonvin's group. And by using external database information, we integrate all of this data into our analysis pipeline that in 2022, we have also integrated, we have started to integrate artificial intelligence also in order to get to our results, which is basically trying to understand how proteins form this kind of complexes and then higher order assemblies and how they look like, what is their architecture. And here I'm showing one of the focuses of the lab, which is the complexes involved in pyruvate oxidation, which are like the mitochondrial complexes of the pyruvate hydrogenase complex, which gets pyruvate and produces acetyl-CoA, and the enzymes of the Krebs cycle as well. And in order to try to understand and apply all of these methods, I have here in Halle a very international lab, and I'm very happy for it, and includes like 8 different nationalities, and everybody's working on the things that I'm showing afterwards. So the first thing is to understand what we see in Cryo-EM and to try to, with our experience, to describe them. So basically, as I told you, we have an organism of interest, we homogenize it, we take the cell extracts, and then with cryo-electron microscopy, we can start seeing this kind of higher order assemblies. So with Anne-Claude Gavin, we have tried to understand what kind of structural organization is this. And from my lab, we started to develop methods in order to simplify a bit this kind of cell extracts using size exclusion chromatography or sucrose gradients in order to separate, for example, the complexes by size and therefore to probe better and more efficiently their content. Also, we are interested in how disordered regions are found in this kind of higher order complexes that can bring proteins together or have other functional roles in these metabolic complexes. And more recently, we are also interested in how artificial intelligence can be applied in the different steps of our protocols in the lab to further understand how the structure of this kind of protein communities look like. So our model organism, as you can see here, is Hitomium thermophilum. Hitomium thermophilum is a very good model organism for structural biology because it is a thermophilic eukaryote that you can grow at 55 degrees of Celsius or more, and it's at the limits of eukaryotic multicellular life. And here I'm showing publications from Heidelberg, for example, where its genome was published, then its proteome was annotated, then here a lot of protein-protein interactions identified for this organism were reported, and here we started performing also activity assays and structural characterization of its protein contents. As you can see here, it's a filamentous fungus with very nice nuclei, and then when you do your classical electron microscopy, you can appreciate that it's full of ribosomes, the small dots, but also mitochondria that you can see here. And this kind of mitochondria are important for us because this is where pyruvate oxidation localizes. So here I'm showing you a picture of a cell extract using cryo-electron microscopy. So we take our cell extract from Hitomium thermophilum and we freeze it, we vitrify it, and then put it on our microscope and acquire such images, where again you can see this kind of interactions and different kind of molecules residing there. So I'm going to be talking about this molecule here from our cell extract. And yeah, so from this molecule, what we do is we have our Hitomium thermophilum liquid culture. We do our cell lysis and size exclusion chromatography of the native cell extract. And then we get a reproducible size exclusion chromatography profile. And in this kind of profile, we are focusing in this presentation that I'm giving you on this kind of first fractions where there are megadalton complexes that elute. So there are from 4 megadalton to 10 megadalton complexes. And in particular, in this kind of fractions, we can see very beautiful signatures inside. For example, like membrane signatures or this kind of rosettes, filaments as well, and smaller liposomes. But also we can see this kind of molecules of 10 megadalton, which are specific structural signatures of how this complex, which is called pyruvate dehydrogenase complex, looks like. So what we do first, in order to understand what we have there in our cell extracts in this kind of fractions, is we are performing kinetic assays inside. So we are trying to see if the complexes that are there, some of the complexes that are there, they are active or not. And as you can see from this kind of plot, the complexes that we are interested in, the pyruvate dehydrogenase complex, PDC, and the oxoglutarate dehydrogenase complex, OGDC, they are active. And these are like very well-known complexes from our biochemistry books. They are alpha-ketoacid dehydrogenase complexes, acetylating the CoA in the case of the pyruvate dehydrogenase complex, and succinylating the CoA in the case of the oxoglutarate dehydrogenase complex. And here also, in the mass spectrometry data, you can see that different oxoacid dehydrogenase complexes like the OGDC, the PDC, another alpha-ketoacid dehydrogenase complex, the branched-chain ketoacid dehydrogenase complex, elute across this kind of fractions. And in addition, with antibodies that we produced, we can even follow this kind of component proteins that, for example, pyruvate dehydrogenase complex has across this kind of fractions. So we really see that in these megadalton fractions, we have not only their proteins, but the full assemblies. So the next step to characterize what is happening inside this kind of cellular fractions is to image them, to put them at the microscope and acquire our images, how they look like. So we know that they are active, we know that the proteins are there, and we can identify them with immunoblotting as well. So this kind of large-scale imaging that we performed is that we are putting each fraction one by one in the electron microscope and acquiring our images from these fractions. And here I'm showing with blue what we think is the pyruvate dehydrogenase complex, PDC, and with red, the oxoglutarate dehydrogenase complex, OGDC. And by doing here statistics on retrieved particles that are being averaged after 2D classification, we can see that most of the particles are not looking with a cubic core or with this kind of symmetric core, but there are other molecules. But the complex that we are interested in with the cubic core and the symmetric core, they show this kind of particle abundances that they might resemble to you what we saw with activity data and with the mass spec data and with the immunoblotting data. So after we collected all of these kind of particles, we do this classification, and then we can systematically reconstruct in every single fraction that our two molecules of interest elute their overall shape from our low resolution negative stain data in this case. And what you can appreciate is that we can reconstruct asymmetrically in this case the cubic core in the case of this complex and the icosahedral core in the case of this complex. And all of this data that I presented you suggests that this structure here is the pyruvate dehydrogenase complex, and this structure here is other types of ketoacid dehydrogenase complexes, either one or mixed. And what you can appreciate from the color coding that I show here is that these kind of structures have not been reported before in their totality. With X-ray crystallography, we have determined, for example, like the overexpressed cores or like at high resolution the subunits around, but how all of these are coming together in this kind of megadalton assemblies, 10 megadalton in the case of the icosahedral molecule, 4 megadalton in the case of the cubic molecule. It was not reported. And we can see this kind of clustering outside the cores. And yeah, as you can see, this kind of clustering is more or less seen across our reconstructions from the different fractions. So after we did this, we wanted also to look at higher resolution, what we have in the fraction and what kind of insights we can get from there. So basically, we wanted to focus here, in this case, in the pyruvate dehydrogenase complex, and we did again our size exclusion chromatography of the Hitomium thermophilum native cell extract, and we collected this fraction 6, where there was the highest abundance of the pyruvate dehydrogenase complex that I showed you. And here we developed more antibodies to directly identify all of its different protein subunits that in total it has 176 of them, of these 5 polypeptide chains. And in the meantime, we have also developed in my lab an in-house activity assay to derive KM values for this huge complex. In this case, we could basically achieve for the core of the pyruvate dehydrogenase complex a resolution of around 3.9 angstroms. And at this kind of resolution, we could basically model inside the density all the 60 subunits that are forming each icosahedral core. And we could basically see that the coenzyme may nicely fits inside its binding site, as well as we could see some kind of exotic interactions like this kind of p-electron networks that arginines from different subunits could form. So, and in this case, because we have this kind of higher resolution of the core, we want to see what is happening inside our core. And what is happening there is that basically in the pyruvate dehydrogenase complex, you have a complex that is being formed by the E1 and the E2 proteins. Then you have in the E2 core, in this kind of core, this kind of transcatylase reaction where the coenzyme A is getting acetylated, and then you have another interaction in this kind of huge assembly where the E3 and the E2 are binding. So basically this complex is composed of basically three enzymes, E1, E2, E3, that act in a series in order to do the acetylation of the CoA and the reloading of the lipoyl arm which exists in the E2. And we wanted to see specifically this kind of interactions in the active sites that are happening inside our core. So basically what we did is something that very simple. So what we did is that we, back then, only less than 1 year ago actually, we modeled with homology modeling our domain, which is carrying this kind of lipoyl lysine, which inserts inside the active site of the E2. And what we basically did here is that we docked this by knowing the biochemical restraint where the lipoylated lysine should insert into the active site. And we try to understand how the interface is, what kind of non-covalent interactions exist, like its bending surface area, and how does this compare, for example, to other kind of transient interactions. What we basically saw is that this kind of interfaces with the Cryo-EM determined E2 core are regulated mostly by ionic interactions and some hydrogen bonds that are organizing how the lipoyl domain might bind. So, and what was interesting is that if you see the electrostatic surface potential maps of the Cryo-EM structure, you can see how nicely this kind of two molecules, they come together for the reaction to be performed, as well as the coenzyme A, how it can fit in its binding pocket with which is like positively charged. So we go back to the complex, how it looks like. So this kind of insights that I gave you were happening in this part of the enzyme, but you saw that there were a lot of additional densities around corresponding to the E1 and the E3 subunits of the full complex, which is composed of the E1, E2, E3 subunits. Another story that we have that I'm not going, unfortunately, to present due to lack of time, but we can of course discuss it, is that we identified another protein inside this huge core, which was a component, a known component of the pyruvate dehydrogenase complex, but ambiguous of its location, and we find it as a trimer inside the core and at low resolution we identified it. But here, what I am showing is basically that we identified with Cryo-EM this kind of additional mega-Dalton compartment on top of this core. So basically, when you look at our class averages from this 10 mega-Dalton enzymatic complex, you could directly see that there are persistent densities that are surrounding this kind of core. And when you do an asymmetric 3D reconstruction of the complex, you see some strong clustering, like a crown on top of the core of the molecule, and some other external densities, weaker densities around. And by doing a lot of computational modeling and docking and fitting and actually benchmarking the significance of the fittings of the E1 and the E3 proteins in all of these densities, we could be confident of the fit of 2 E2s and 1 E3 in this kind of corresponding densities, whereas in the other densities, the fits were not significant for either of the two. So in this model, what we present is basically the first model of how the E1, the E2, and the E3 are spatially organized. And here, what is very interesting to see is that from this kind of model, we can derive actual distances between the E1, the E2s, and the E3s of the full pyruvate oxidation reaction. As I told you, but I didn't cover in my talk, we identified another protein inside the icosahedral core of the E2 protein, and we also saw surrounding this kind of molecules here, this kind of density, which is surrounding our enzymes. And what we think is that basically these enzymes are in proximity by other E1s and E3s that are forming this kind of nanocompartment. And the lipoyl domain, which basically is the arm that is bringing the substrate from one enzyme, the product from one enzyme to the other as a substrate, and then again to a product, this whole reaction is happening structurally below this kind of nanocompartment. And overall, what we saw from this reconstruction is all of the different kinds of distances between active sites, and like we could understand, like for example, how big flexible regions are, because these are the ones that are organizing the E1s and the E2s, or the E3s and these other proteins, the E3BPs, in proximity. And overall, we could understand structurally this kind of reaction. Moving from this molecule, what I want to show you here is basically for this kind of cell extracts, how we developed together with our collaborators in EMBL Hamburg, Gregor Czonowski, a protocol to try to understand more structures from the cell extract. So what I showed you is reconstructions and a bit of structures from cell extracts, but only of one molecule, the one on the bottom here. But the idea is to move towards simultaneous characterization of this kind of protein communities and their members, how they look like, and how we can use artificial intelligence for that. So again, here I'm showing the biochemical data, which are very important for the interpretation, because of course we need our reproducible gel filtration profile to get the fraction of interest where we want to do our further analysis. Abundance data from mass spectrometry, what kind of protein complexes we can identify in this fraction. Also activity data, if our molecules there are active, because from preparation to preparation, activity might change because of a minor step in handling. And also what we see is that our proteins are also there with immunoblotting data. And after we do this kind of biochemical characterization, we can pick basically all the particles that we are acquiring in our images and derive 2D class averages. And what you see here is this kind of different class averages from 4 distinct signatures. And here, after knowing, I will show you in the next slide how we know what protein complex, which protein complex is what. You can see that this kind of signatures actually represent 10% of all the particles that existed in our fraction. So also here you can see this kind of flexibility that persists in this kind of class averages. For example, this kind of cubic core is surrounded by diffused densities. This kind of icosahedral core is surrounded by less diffused but still diffused densities. And this kind of symmetric hamburger shape, you have this kind of additional densities around, and this asymmetric density, again, these diffuse densities. So by having this kind of class averages, we can ab initio reconstruct these shapes, and you can see without a reference, we can recapitulate a bit their symmetry. And in the case of signature 4, the asymmetric map. Here, what we did is something that very interesting, is that the PDBJ, the PDB Japan, has a tool which is called Homology, and this tool, what it does is something quite important, that given a map, it cross-correlates your map to the most, to the whole EMDB, which has deposited electron microscopy maps, but also to PDB structures and their shape. So this kind of shape cross-correlation, for 3 of the signatures, it worked. So because we knew, because there were structures of similar shapes and of homologous organisms in the EMDB and PDB, we could basically annotate the 3 signals. So in the case of the cubic reconstruction, we could see that it is the E2 core. In the case of the icosahedral, the pyruvate hydrogenase core. In the case of the dihedral, the hamburger shape, the fatty acid synthase. But in the case of the asymmetric reconstruction, this kind of tool failed, and we had to go back and look at the hits that it retrieved, and by back projecting the structure to see with the back projections which is could be this molecule, and finally we saw that it was the immature 60S eukaryotic ribosome, and on the right you can see the projections from the pre-60S of the bacterial counterpart. So we could basically refine this kind of maps that we got de novo at decent resolutions, where we can see features that correspond to the calculated resolutions for the maps. And actually, what we developed with Gregor Czonowski from EMBL Hamburg is something very, very interesting that I'm going to show you here and bring you step by step. So we have our native cell extracts maps, right? This kind of 4 maps. In the case of fatty acid synthase, we clearly know that it is fatty acid synthase. In the case of pyruvate hydrogenase, we clearly know that it is this molecule. In the case of the pre-60S, we clearly know that it's this molecule. But here, for this kind of map, because different protein complexes inside the cell can have this kind of cubic cores, we had 2 candidates that at this resolution of 4.5 angstroms, we didn't know which molecule fits in the density. So in this kind of approach here, we are trying to answer this. So in the first step, we have our native cell extracts maps, and in the case of the cubic core, it is still unidentified. Then what we do is that by using collab fold, we can derive the trimeric models that can fit here from our candidate proteins. And then we can use our normal tools as structural biologists Scoot and Phoenix in order to refine the fit of both of these models inside our experimental map. After doing this, we are extracting only the backbone and then using the tool from Gregor Chonovsky, which is called Find My Sequence, and it is another machine learning algorithm that basically guesses your sequence based on the Cryo-EM map. We could, by using this kind of tool, to unambiguously identify that the molecule that fits there is the oxoglutarate dehydrogenase complex E2 protein and not the branched chain ketoacid dehydrogenase complex E2 protein. So here in this case, AI helped us not only to get very good models for to refine in our maps, but also it helped us to unambiguously identify which is the correct model. So what do we learn from this kind of exercise? So first of all, we have some beautiful structures that are refined experimentally from AI models, but also we can see some things in this kind of structures. So first of all, for the pyruvate dehydrogenase complex, the AlphaFold model predicted this kind of backfolded helices as very nice folds on top of our E2 core, but in our experimental density, there is no density for those there. So these things, these kind of alpha helices that are predicted by AlphaFold, they are either disordered in the native endogenous structure of the pyruvate dehydrogenase complex, or this kind of helices are much, much less abundant compared to this kind of flexible states. And that's why we don't have very good, not at all actual density there. Also, the AlphaFold model could fit very nicely, of course, the overall densities that we captured, but some additional densities inside the core were not covered by the AlphaFold model. So it gives us certain hypotheses what could be there, and all of these things that we answered for this E3BP protein that I didn't present you here. The next molecule that we modeled with CollabFold was basically the fatty acid synthase from Hitomium thermophilum. And here you can see that before, when we submitted actually this manuscript, that the tools were not available yet online, we did homology modeling. And with our homology models, in this kind of low resolution density here, what we could see is that this domain, which is the acetyl-CoA binding domain of fatty acid synthase, compared to the yeast structure deposited in PDB, we see some kind of rotation of the domain, which is probably mediated by a flexible linker. And during revisions where we included this kind of AlphaFold calculations in our paper, what we saw for this molecule is quite interesting. So first of all, AlphaFold did not learn from protein complexes, but as you know, it learned from around 160 residues batches of structures, and it could actually very nicely recapitulate a hexameric interface in the core of fatty acid synthase that we only refined just a little there, but still it predicted the acetyl-CoA binding domain of fatty acid synthase in the conformation that it was crystallographically determined from yeast and not in the experimentally seen conformation of the endogenous fatty acid synthase from our native cell extracts. And the next step is the oxoglutarate dehydrogenase complex. So what happened here is what I told you, that for this cubic core, we couldn't decide if the core corresponded to the oxoglutarate dehydrogenase complex or the branched chain ketoacid dehydrogenase complex, because both of these complexes have cubic cores. And what we saw here is that by just simple cross correlations, we couldn't differentiate which is the best fit. So we used this protocol that I showed you before, and we unambiguously identified that it corresponds to the oxoglutarate dehydrogenase core. And here I'm showing you the predicted AlphaFold model. Another interesting part in the predicted AlphaFold model in this case is that, of course, the model fitted very nicely in the map, but again, an additional domain of this protein was found to be ordered and bound to the core. This kind of domain, which is the lipoyl domain, which carries the lipoyl from one enzyme to another, this arm that these dehydrogenase complexes have. And again, in our density, it's not there, it's absent. And actually, in our experimental data, what we see is that these external subunits of the E1s and the E3s that I explained before, they are very, very diffused, which means that the conformation that AlphaFold predicts for our E2 protein, it's again a very minor state and very transient interaction that cannot be captured in our experimental data, but of course it provides a very valuable hypothesis on how this kind of reaction might take place. So what I presented to you today is basically some results from our 3 papers from the lab, where in the first paper we derived the full higher order structure of the pyruvate dehydrogenase complex. In this paper, we did this protocol that I showed you last with characterizing these different structural signatures with Cryo-EM and artificial intelligence. And I also showed you parts from this paper where we have this higher resolution of the core of the pyruvate dehydrogenase complex and how we understood better the transcaterylase reaction happening there. And yeah, we now moved as a lab to develop our tools and collaborate also with the MBL Hamburg to somehow integrate in our Cryo-EM pipeline artificial intelligence to study native cell extracts. And we are very happy that we got the covering structure and also a very nice preview written by the Gatso-Yannis group. And overall, what I want to tell you is that we use in my lab Haddock, but we also now try to use AI algorithms to understand how proteins interact, also what Capri is doing all of these years, and I also participated in that. And by understanding this kind of binding using energetics to include experimental data like electron microscopy data to reconstruct this kind of higher order assemblies, megadalton assemblies. And these kind of cell extracts, they fit into the bigger picture of protein-protein interactions of the cell, whereby taking a picture of cell extracts, basically what you see is a snapshot of the protein communities residing within it. And by acquiring more and more data, more and more images of cell extracts, you can see this kind of interactions, higher order assemblies present there, and try to average and increase the resolution in order to understand how they are structurally at higher and higher resolutions. And hopefully we can map all of these interactions and add the experimental dimension to the interactors. So at this point, my presentation has come to an end. I would like to thank all my lab members and also all my collaborators that with them this work, of course, without them this work would not be possible, and also my funding agencies, especially the Ministry for science here in Germany that funded my microscopes and several people from my lab. And thanks a lot for the invitation and the opportunity to present here.
Thank you, Panos. This was absolutely exciting, really, really, really exciting. I must say, so hundreds of questions, but, if I may just ask, one question first, and then I'll ask the others. Did you find any, the idea that metabolic enzymes that are in the same pathway, kind of form a complex is very old?
Yes.
Right? Substrate shuttling. That was the idea. Do you see any evidence for that? In other words, do you see these distances between the different subunits, the substrate to product, proximities? Do you see any evidence that could explain this older theory of substrate shuttling?
Not yet. So what we see is for sure from the proteomics data, we see enzymes co-eluting in the same fraction, in the megadalton fraction. For example, we see all the enzymes of the Krebs cycle in the first megadalton fractions reproducibly.
Yeah,
so all of them are there. This is the first insight that they must be in at least in proximity to elute together. The second thing is that in the case of the complex that I showed, these complexes are brought in proximity by disordered regions, but then the substrate shuttling is happening through a flexible arm. So there is no like direct channels that are forming between the enzymes, but there is an arm which is bringing the intermediate from one enzymatic site to another. But this thing does not exist in the enzymes of the Krebs cycle. So there we don't have images yet, but we have at least like this kind of data that do not exclude substrate channeling.
It could mean something different, but this was a concept, of local concentration, and somehow that the substrate will kind of move along, the surface of the protein and not be solubilized or somehow.
Exactly. So we don't have direct evidence for that. So exactly, this is what Srer and Lippmann used to write, of course, yes. But we don't see something like that yet. But the more images we acquire, the more we are able to answer this kind of questions.
Wonderful. So other questions, others, please, do ask Panos. I think we have a little time if you're not too busy. I think we should ask.
Sorry that I was a bit.
It's okay. It was so interesting. So, we definitely are happy that you managed to show us everything. So go ahead. Okay. I have more questions. Anyway, the other question I wanted to ask is the less populated, images that you had of the particles, were those, basically portions of the bigger complexes? In the case of the mega complex, the most populated was the mega complex, and then you had these other, with these different symmetries. Were this already part of the complex, of the bigger complex? Were those where you had these predicted regions that were predicted as really structured by AlphaFold, but then they were, in your images, they were very flexible.
Yeah, all of these are part of the same complex, yes.
Okay, part of this, of the bigger complex.
And of the protein itself.
Yes. So you see that there is a whole theory of how the mega complex would form. In other words, the parts that are really very flexible, you have a core that will form, then you have a flexible part that actually is flexible without the presence of the other components, and it becomes fixed once the other components are there. So do you see these fixed, predicted regions in your core in your mega complex?
No, they are still flexible. So we don't see any density connecting the fixed.
In the other complex, not Not in the mega complex, in the big mega complex from the same subunits.
Shoshana, just ask me the question differently because I don't think I studied 100%.
You have a mega complex,
yes,
and you have components of the complex, you have the core. Then you have these smaller complexes that you try to identify. Are those part of the mega complex as well? I mean, these
exactly,
are the
yes.
So, but you're showing that in these smaller complex, in these portions of the bigger complex, you have flexible regions.
Exactly.
Which are actually predicted to be structured by AlphaFold.
Yes, yes.
Now, what are they? Are they structured in the mega complex or not? Or they remain flexible in the mega complex?
Some of them, they are folded, like for example, the lipoid domain. So this is folded, but it moves around. And the other helix that I showed, which is not there in our reconstruction, it must not be there because it hides the active site. So the reaction cannot happen because the lipoid domain cannot enter.
Okay, so you have a conformational change. In other words, when you have these components that probably are, formed, there is a hierarchy in which probably the mega complex forms. So there must be something, some conformational change that happens for other subunits to bind. Either you fix, the disordered regions, or you change conformation, something like that. That would be interesting to see.
Yes. So this is a follow-up work that we are currently doing and preparing. So there are like specific regions in the disordered linkers which fold in order to accommodate this kind of subunits around. I can show you. So, and they share a lot of common features. So they can undergo this disorder to order transition to bind the external subunits.
You fixed now.
So this is what is happening. So basically, I can go to this slide here. So here, in this kind of reconstruction, this kind of subunits, the E1s and the E3s are held there with disordered regions, and these disordered regions from the E2 core, these are extensions, disordered extensions of the E2 core. They fold in order to accommodate them, and so this is what we are trying now to understand, actually. But not all of these disordered regions are occupied with these molecules.
Anyway, I'll talk to you because this is an aspect of the conformational changes that these proteins are undergoing when they interact. This is really a very interesting, interesting angle of the analysis.
I agree. It's something that we are pursuing now. So now with AlphaFold, actually, it's something which AlphaFold can predict this kind of bound states. And we are trying to compare the models that we get with AlphaFold, how similar they are. Because this is like this ordered conformation that binds these E1s and E3s is not known. So we're trying to understand how this folds in different species, and we do this kind of species comparison. And we see that for the different keto acid complexes, the fold is similar that you get there. But we are now looking at that. Yes.
Well, thank you. There are other questions in the chat. I'll read them for you from Ruslan, also in electron microscopist expert in our department. Do you know how strong protein-protein interaction needs to be to survive your purification protocol, including cell disruption?
Okay, you dilute.
Obviously, not completely strong because you get the different portions, right?
Yeah, exactly. We get the different portions, Ruslan. Thanks a lot also for your question. And so basically you dilute when you do the lysis, of course, you dilute like 10,000 times. But then what we load in the column is like 30 milligrams per ml, which is like around 12 times more diluted than what is inside the cell, right? So we lose for sure interactions that are dependent on such ranges of on and off rates. But yeah, for example, like we have much less signaling molecules and signaling complexes in the fractions and much more metabolic complexes. So we lose this kind of trans interactions, probably the micromolar ranges or like millimolar ranges for sure. These are the ones that we lose. But then when we reconcentrate in order to inject the column, we might get some again. So yes, these ones we lose, but still we retain a lot of interactions of the higher order. And these ones, they are stronger, but they include weaker interactions. For example, I showed you that we can actually know where this kind of lipoyl domain might bind, and this has extremely transient interactions. But because it is like, it has a flexible linker covalently bound to the core, we can maybe capture it. So this is the answer. I hope you are satisfied. So what kind of column we use? So we use the BioSep S4000 column. So it's a column which was first published in Nature Methods in 2012 for separating large molecular weight complexes. We benchmarked it also in EMBL. I benchmarked it to retain this kind of cellular complex because it's the only column where I can get reproducible fractionation of complexes up to 10 megadalton. So that's why we use this specific column, and we have a trick. We connect this column, which is an HPLC column, to an FPLC machine. So to get advantages from both methods.
So there was one other question here. Did you get reasonable 2D classes of any other big complexes besides the structures you have shown? How difficult would it be to try to solve structures of the other complexes that you have highlighted in the micrograph? In other words, did you look at everything?
Yeah. So Ivica, thanks a lot for the question. Yeah, this is like something which requires a lot of discussion. What I want to say only is that right now we have up to like 5 simultaneous characterizations of like protein complexes or like parts of this kind of bigger complexes. We cannot get more, and we don't know why is this. So maybe it is because we don't have enough particles. Maybe it is because we need to do tomography of cell extracts to get initial shapes in order to refine the shapes, because we lose with single particle 3D information, because we cannot reconstruct from the 2D averages the 3D, because we have a lot of different 2D averages. And then when we are trying to reconstruct, we don't get useful maps. So it could be that also like the algorithms right now that we use for 3D reconstructions in Cryo-EM are not ready for such heterogeneous samples, and that's why we resolve only 4 or 5 of them and not like many, many more that we have. So it is, I think, very difficult right now. And but if we had here a faster microscope with a faster camera, we could acquire like 10 times more images, and then we could go deeper and deeper into the structural characterization of cell extracts and what is there. So it also has to do with your primary data, because we derive this kind of results that I showed you for the 4 different complexes with 3,000 images with pixel size of like 1.5 angstroms. If we used 30,000 images and pixel size of 2 angstrom to get 4 angstrom reconstructions that we can efficiently like say what is what, then we could get more maps. Nothing, Ivica, of course.
Okay, people are happy. Any other questions? Anastasia, do you have questions? I think no.
Well, I have kind of a naive question, because So when I saw your reconstruction of like the satellite parts of the core complex, I mean, you said that you run many different simulations to identify them, but I'm just wondering how that works, right? So you probably need some kind of hypothesis to start with, and then you dock the structures into density. So yeah, so I mean, I imagine it's kind of a brute force approach, but I can't really, yeah, I'm just wondering how that works.
Okay, yes. This is also another excellent question. Of course, this is one of the weaknesses of low resolution models, that when you have a low resolution density, you are not sure if your protein fits there or not. So the hypothesis that we had were the following. In the external densities, you have only E1s and E3s and nothing else. So only these two proteins are there. And this is backed up by the mass spectrometry data. So we find them there. And we expect them to be in the outside densities. So the second thing is that we make a model of it. And there are crystallographic models of very high accuracy of yeast, and Hitomium has very similar sequences. So we got how this kind of proteins look like. And then what we did is that we systematically low pass filtered them to 5 angstroms, 6 angstroms, up to like 30 angstroms, and fitted them within each other's densities. And so when the fits are wrong, and at which kind of thresholded resolution. And we saw that the fits were wrong after 25 angstroms. Our map was 20 angstroms. So when we fitted them 10,000 times each in our map, and they clustered one of them in one density and the other one in another, then when we saw these clusters and the fit consistently there at 20 angstrom resolution, we said this is the correct fit. But of course, yeah, this is a computational work. It also depends on what kind of thresholds you use and generally. So this is how we did it. And for me to put them in a paper, I'm very confident that these are the places where the 2E1s and 1E3 binds. And that's why we didn't show all the other fits around, because we were not sure. Okay. So this is what we did, how we did it.
Yeah. And I was wondering, looking at those data, I was wondering if a different approach could be, I mean, you show that you could go from a density with machine learning, you can go from a density to a sequence, but what if you start from a model and that you kind of predict the density, that you could see a different resolution from a structure? Wouldn't that be like a faster way to maybe to map images to expected densities?
Now we go to the invited proteomics paper from Shoshana. So this is what we are trying to do there. So what we want to do is, we have the proteomics data, we have a network, we see some proteins clustering in the network, we produce the AlphaFold models of their interactions, and by knowing the stoichiometries from the protein data bank to reconstruct a model, low pass filter the model, use it for picking Cryo-EM micrographs. Mm-hmm. So this is the idea, exactly, like Anastasia, this is exactly what we are trying to do. So it's something that might or might not work, mm-hmm. but it's another application of possibly AlphaFold to try to understand our reconstructed densities and what we have in our micrographs, actually.
Yeah, fantastic, great. Yes. Thank you so much. Other questions, guys? Really fascinating. So, you know, you're going all the ways on different sides, and using all these tools will come together, and this would be fantastic.
Exactly. And I really believe that this kind of energy calculations that we do in docking and in scoring and everything, they are going to come together with the AlphaFold predictions, because it's something that AlphaFold has not exploited yet. No. And it's, I think that it's something that, yeah, it's going to come together, as you said, Susanna. Yes.
Great. So thank you so much for this fantastic talk, and all the best for continuing work. And we are, expecting your wonderful manuscript at some point.
Hella, wonderful, I don't know. It will be good, but yeah.
Okay. Yeah. Thank you for everybody, and thank you, Panos. And I'll write to you about the conformational changes. And this is something that I'm kind of very interested in having examples. So I'll write to you.
Yeah, Susanna, also we can also discuss about the binders and the non-binders in the case of these kind of huge complexes.
Yes.
So basically, the discussion that we had before we go is that in this slide here, do you see my screen?
No.
Okay. So in this slide here again, you see you have this kind of external subunit. So the E1 and the E3 are in proximity, but should never form an interaction interface. So these are two proteins that should not interact with each other. So this is a pair that we know that is a negative interaction.
Yeah. And you know that there is no other density that is kind of hidden, and it's the entire sequence is in there.
No, it's not. It's only the folded domains. So
you have these ordered regions which still could eventually interact.
Everywhere.
Yeah. Okay. So this is okay.
Yeah.
Okay. Maybe we can talk about this, because it's really fascinating to see what's going on. Okay. Thank you.
So, Susanna, thanks really a lot for the invitation. Thanks all of you for your questions and the insights. And yeah.